I will very quickly introduce myself. I am Vincent Noel, professor at the University Paris-Cité at the Department of Chemistry. And today I would like to speak about functional inks for smart label design. So there's, due to the expanding, the expense of connected sensor and biosensor, there's a requirement to secure the data which are transmitted. So there's a need of secure management of sensitive personal information and to guarantee the secure identification and authentication of data transmission and storage. So today, there's two main approaches to secure such operations. This is asymmetric cryptography, such as this is algorithms, so information security by levering the computational complexity of factoring large integers. So this is very strong algorithm, but today the complexity becomes surmountable with quantum computing and also due to machine learning. So there's another kind of insecurity for data. This is symmetric cryptography. So the entities share private keys, typically stored in a non-volatile memory for encryption and decryption of messages, but that's not so interesting for connected sensor and for IoT devices. And moreover, the stored key can be compromised by physical and side channel attacks. So that's why today there's a lot of strong interest in another way to secure the transmission and the data. And the idea is to use a physically unclonable function. So in such devices, the key is stored stored physically on the devices. And so the devices just read a kind of physical signal. It could be magnetics, electronics, or radio frequency. And there's another kind of physical signal which could be very interesting is optical signal, because optical signal can be unique. There's a high output complexity of optical signals, and what's also interesting is that there's a resilience against erratic devices for supply. But the challenge is for optical physically unclonable function is to have a high encoding capacity and ease of recording the signal. This is, what does it mean? It means strong light emissions. So the idea is to store the key inside materials which have optical properties. So today, the most investigated materials is quantum dots, so fluorescent tags. This is really interesting materials because you have a very high fluorescence emissions. But this is fluorescence, and there's some drawback to this kind of signal. And the most one is photobleaching, meaning that the emission decrease as when you interrogate it, as a decrease of the fluorescent signals. And in another field of research, other tags are really useful for imaging, for bioimaging. This is our Raman tags. So Raman tags are based on molecules, and this is very specific vibration of chemical bonds. And so you have a spectroscopical signature in a specific wavelengths here of wavelengths, and you have very sharp peak. So this is very interesting signal that can be used for key security. But the problem is that you have a low emissions of this kind. This is a very low emission spectroscopy. So you need to enhance the signal for the target applications. But Raman tags are really interesting compared to fluorescence probes, because as you can observe on this comparison of spectroscopic fingerprints, fluorescence are broad peaks, where Raman are very sharp one. So that's really interesting. So multiple sets of narrow peaks, a low spectral overlap between the peaks, so a high multiplexing ability. And you have a single source light, which it is very easy to record a Raman signal. You just have to excite it at a given large wavelengths, and you obtain this kind of signature with negligible photobleaching compared to fluorescence. But the problem is that the signal is too low. So in the PRINTUP Institute, we developed some ink based on Raman props graphed on plasmonic nanoparticles, because thanks to the plasmonic effect, you can enhance the Raman signatures of molecules, which are very fingerprints, spectroscopical signatures, and you can enhance the signal very high. You can reach 10 to the seventh amplification of the cell signal thanks to the plasmonic